Hello and welcome back again. My name is Jess and I'm your representative from the Antibody Society. I hope you've been enjoying our interviews with researchers behind the images that you submitted for our 2025 calendar. Make sure you visit us at one of our booths and pick a copy up or download one from our website. Today I'm joined by our March edition, Andy, who is a fellow researcher at the University of Southampton. Welcome, Andy. Thank you so much for submitting your image to the 2025 calendar. It is an absolutely stunning image that we have behind us. Would you like to introduce yourself and tell us where you did the work for this?
Yeah, thank you very much for the compliment on the image. So the image came from when I was doing my PhD and postdoc at the University of Sheffield in the Sheffield Hearing Group. And it was a kind of side project that came about from the main research that I was doing over in Sheffield. And I wanted to have a look at the inflammatory response within a transgenic mouse apical coil of the cochlea. And yeah, I wanted to see how that looked. And this was one of the images that came out from those experiments, which came out a lot nicer than I was expecting.
Yeah, it's got an amazing structure to it. I'm looking forward to learning some more about it. So where you're based at the University of Southampton now, how did you sort of get to swap from cochlea to neurons, I believe?
Yeah, so my original project was obviously hearing loss, and then the side project where I decided to look at inflammation kind of sparked my interest in the inflammatory responses. I also tried to then look in the brain as well at the neuroinflammation following hearing loss in these mice. And from that, I decided that neuroinflammation was an area of research that I wanted to pursue. So that led to me changing out of hearing and pursuing the position I'm in now, where I'm working in neuroinflammation, working with stem cells and iPSC derived cerebral organoids and microglia.
Brilliant. Well, inflammation in any site is highly relevant to the people who are part of our society. So I look forward to learning a bit more. So do you want to show us a little bit behind the research that you did that generated this image?
Okay, so this is the one of the main figures from the paper that I was doing or came out of my PhD during my time at Sheffield. So the research was centered around age-related hearing loss, and also we used transgenic mouse models of hearing loss. And in age-related hearing loss, there is a rewiring of the efferent system of the cochlea relating to the hair cells. So within the cochlea, you have inner hair cells which allow us to hear, and you have outer hair cells which amplify sound.
Yep.
And it's the inner hair cells that undergo this rewiring with aging, but also if a process called mechanoelectrical transduction is disrupted, then we found that this reinnovation takes place, but over a much shorter time span. So rather than having to use aged animals, we could use much younger animals. And
Okay.
this allowed me to look at mechanistic changes that took place during this time point.
Yep. So what
So,
age are the animals that you've used in these experiments then?
so they spanned from, so the kind
Across
of
the bottom, yep.
developmental animals we used were P9 and P10.
Mm-hmm.
And this is when the innovation is still maturing and developing, and the inner hair cells are innervated by these efferent connections. And the efferent
Okay.
connections within this panel are indicated by SK2, which is a small conductance calcium activated potassium channel, and is expressed by the inner hair cells. And I used chat to label the efferent fibers. And where it's white towards the base of the inner hair cell, which is labeled in blue using myosin 7A, that forms a functional synapse. And what we saw is at day 24, the efferent reinnovation hadn't taken place. However, the mice had fully matured their developmental innovation. So within that sense, you would lose the efferent innervation of the hair cells, of the inner hair cells, by day 21 in these mice. And you can see
Yes.
that in the wild type mice here in B, there's no expression of either SK2 synapsing with the chat. And in the mutant mouse model, the myosin 7A, you can no longer see the hair cells because they no longer express myosin 7A. And you see that only this one cell in the row of around 9 or 10 inner hair cells expresses the efferent fibers. So it's on their way to losing. Yeah.
Yeah.
So, well,
how do you score the hearing level? Would these score high on a hearing loss score, or is it sort of moderate hearing loss at that stage?
So at this stage, at day 24 to 26, they are starting to have a threshold shift in their hearing.
Okay.
And these mice become profoundly deaf around 35 days old.
Okay.
Which is shown within the paper. So by this, at day 24, we're showing that just before the hearing loss is starting to shift, you're starting to see some changes in the efferent innervation of these inner hair cells.
Yes.
And then by comparison, at day 39, when these mice are profoundly deaf, you see a lot more of the inner hair cells have undergone this rewiring and have re-expressed the SK2 channels and have functional efferent synapses on them, indicated by the percentage of inner hair cells that express the SK2 channel and also the number of puncta increasing over time.
So by comparison, in your wild type mice, as they age, do they eventually lose the inner hair cells as well? So eventually they would look like that, just your transgenics have fast tracked this.
So this is in the sense of the inner hair cells haven't died.
Yeah.
It's just the marker I'm using, the antibody I'm using is no longer expressed.
Okay, so they've changed their phenotype.
Yes, so there's electrophysiological changes that the cells undergo during time in this transgenic mouse model, as well as morphological and synaptic changes. So this mouse model here, this myosin 7A, is replicating a form of human hearing loss called Usher syndrome. So myosin 7A is one of the proteins involved in the process of mechanoelectrical transduction, which allows us to convert mechanical stimuli into electrical signals to be sent to our brain. And if you don't have this protein or it's mutated, then it can often lead to Usher syndrome, which has different severities within humans. So yeah, this was one of the reasons why I then progressed to the main image that I submitted, because I was thinking, if there's changes going on with rewiring, then there must be changes going on within the environment.
Yes.
Which led to looking at a marker of the environment and looking at macrophages. So this was a day 35 transgenic mouse of the same strain, the myosin 7A, myosin 15 Cree. So this mouse was profoundly deaf, and there is no other, that's all that's happened to it, that I've not induced any trauma to it or exposed it to high levels of hearing. And we saw this enhanced response with macrophages.
Okay.
So for this, I used IBA1 to label macrophages.
Okay.
And I used TUJ1 to label both afferent and efferent neurons, as opposed to in the figures on the left, where I just used chat, which labels only the efferent neurons.
Okay. No, interesting. So in terms of immune cells, are macrophages the main immune cells that are present at the cochlear site?
Yeah, so they are the most common. There are other immune cells within the cochlea, but at much lower levels. There aren't many resident immune cells within the cochlea. For quite a long time, there's a dogma that it's thought that the ear was immunoprivileged and didn't have any immune cells.
Yes.
Which was eventually disproven, and there are tissue or resident tissue macrophages found within the cochlea.
Yeah. So they sort of maintain a barrier, don't they? So what type of changes promote other immune cells coming in, and like in hearing loss, do you see any of those other cells, or it's predominantly your macrophages in the hearing loss state as well?
So it's not something that I got to look at too much. I only
Okay.
focused on the macrophage side of things.
Yeah.
But it depends on the type of hearing loss or the type of trauma that has taken place as to whether they arrive. So typically you
Yeah.
have, or I can show here,
Oh,
these
cool.
are generally the
Hmm.
locations where you will find macrophages. So the
Yeah.
image I've shown is of the actual organ of cochlea here, and is
Yes.
also showing some of the spiral ganglion neurons. Um,
Mhm.
so some of the macrophages that you would see around the neurons are going to be coming up here through the osseous spiral lamina, and you will also see some in the basilar membrane. And what you can't see in the image is the stria vascularis, which has all the blood vessels. So that's where you're going to see a lot of the resident macrophages. And with
Yes.
aging in particular, the stria vascularis can become dysfunctional, and then you will see potentially inflammaging, so chronic low levels of inflammation within the cochlea, where you'll see increases in macrophages, but in also other immune cells.
Yes. So is there any sort of, it's labeled as cochlear macrophages. Is there anything distinctive about them that sets them aside from other types of macrophages or like phenotypic markers that are unique to them?
Not that I believe so from the research at
Yep.
the moment. I thought they form from the yolk sac, and then they transition down, and then once you get a population within the cochlea, they kind of survive. And if you have any inflammation or any trauma, then they will recruit more via the blood vessels, and also trigger your typical inflammatory responses.
No, brilliant. No, that's really interesting. I haven't heard much about cochlear resident macrophages, so it's cool to learn a bit more about them. So how did your work progress? You mentioned earlier that some of the research for this has been published. How did these images impact on the hearing? Can we learn a little bit more about it?
Yes. So in the context of the paper, we
Mhm.
didn't get to explore any more of the immune responses. We did find some interesting results that weren't included in this paper, but might be included in future papers. So I can't go into them in too
We'll
much
keep
detail, really.
posted.
Yeah. Um, but it's an area of research within hearing that hasn't been massively explored as opposed
Yeah.
to other aspects of hearing. Um, and what I found quite interesting is, especially in response to this mouse model, I self-termed it a model of sterile inflammation, in the sense of I didn't inject anything or cause any trauma. It's just the transgenic mouse model losing the expression of myosin 7A, becoming deaf, and then having this kind of
Yes.
increased immune response that lasted for almost a year, as opposed to typically within the ear, it'll last 7 days, maybe 2 weeks, depending on the level of trauma caused.
Yes.
Um, yeah, so
Oh,
I can't say too
interesting.
much about.
Yeah, no, that's fair enough. So this is obviously relating to age-relating hearing loss, but I know, given we're the Antibody Society, that one of the side effects that you can get for having monoclonal antibody therapy, commonly treating cancer and those sort of related rituximab type antibodies, that one of the side effects can be hearing loss. Do you think this is linked at all to the work that you've been doing in perhaps the macrophages that you've shown, given there is a potential for these antibodies to interact with these cells within the cochlea?
It could be possible, again, due to the amount of inflammatory research being performed within the cochlea. There's not loads of data. A lot of the literature at the moment is saying that the monoclonal antibodies are causing or are toxic, so they're most likely going to kill the hair cells, or they might
Yes.
damage the neurons. But like you say, they could be interacting with the macrophages and the immune cells, and whether that causes some cascade effect that leads to the pro-inflammatory pathway becoming activated, and actually the macrophages removing these hair cells or the neurons when they shouldn't. I don't
Yeah.
believe there's too much research for that at the moment, but hopefully in the future that will become investigated more, because like you say, the downside of the monoclonal antibodies often being used to treat cancer is, for some patients, unfortunately, they lose their hearing.
Yeah, no, that's wonderful insight. Thank you so much, Andy, for that wonderful explanation of your research. And thank you all for tuning in. I hope you've been enjoying this research interactive series brought to you by the Antibody Society, and see you all in the next episode. Thanks.